Hi everyone. Thanks for joining us today for the latest in the SimSip Scientific webinar series. As usual, everyone's on mute as they come in. What we'll do is we'll have the presentation as usual, and then we'll have a time for questions at the end. So please do feel free to engage in that either by chat, using the chat feature, or raising your hand, and then we can discuss your questions at the end. So today's presentation will be by Luc Rougier. I hope I've done that right. Sorry, Luc, if I said your surname wrong. I normally check before we set off, but his paper is well, his presentation is linked to his recent publication, and it's to do with time-dependent inhibition, focusing on the disconnect between in vitro and in vivo for 3A4. If you just want to hit the next slide. So Luc is a director at Eli Lilly in the quantitative clinical pharmacology and discovery PK/PD group. So you've got extensive experience in experimental drug metabolism and inhibition work, and he works in the improvement of in vitro to in vivo modeling translation strategies. So we're thrilled that Luc agreed to present today. We read his publication, and it's one of the key topics that we still there are gaps in understanding on how we do time-dependent inhibition and relate the in vitro data to in vivo. So it was really interesting based on his publications in this field, and the recent one based on linking that together to the predictions of in vivo. So thanks for agreeing to present today, and I'll pass over to you to describe in detail how you've looked at this problem.
Thank you very much, Oliver. And yeah, you did a great job with my name. Thank you. And thanks for the invitation to come and speak today about some of the work that we've been doing to understand the disconnect in the in vitro Cytochrome P450 3A4 time-dependent inhibition and how we translate that to the in vivo. So I want to start by defining time-dependent inhibition. So it's the change in potency of an inhibitor with respect to time. And what that tells us is that at time 0, you'll see a particular potency, but as you incubate with time, it'll take less concentrations of your parent to see that same type of inhibition. This can occur in one of two ways. The first is what we call reversible time-dependent inhibition. And that's where your parent molecule is formed into a more potent direct inhibitor than the parent. And an example of this, or examples of this, is amiodarone and itraconazole. And these are metabolized to N-desethyl amiodarone and hydroxyitraconazole, which are equally potent, if not more potent, than the parent. And so as the parent disappears in systemic circulation, it appears that you are getting more potent inhibition, but it's actually your metabolite that's being formed. Now, these metabolites are reversible, meaning that they exit the active site and then can come back and inhibit competitively into your enzyme. Now, that's conversely to an irreversible time-dependent inhibitor or a mechanism-based inhibitor. And this is where the parent undergoes a catalytic transformation in the active site. And prior to releasing a reactive species, that species irreversibly inactivates the enzyme, either inside the active site or near the active site. From the diagram below, you can see we have a drug and a competitive inhibitor, and much as the name suggests, it competes with the drug for the active site going in and out. However, it does not change the amount of enzyme pool that you have within the system. However, if you have a time-dependent inhibitor, what happens over time is that you reduce that pool of enzyme. And it's important to be able to characterize and understand that, because as you eliminate the pool of enzyme, you eliminate how much enzyme can then metabolize your drug of interest. However, it's important to understand the kinetics of this and the rate at which it changes. If it happens very slowly, and you can synthesize new enzyme, then that might not be a risk. But what can happen with time-dependent inhibition is you can have drug-drug interactions, and more specifically, pharmacokinetic drug-drug interactions, where one drug interferes with the ADME properties of another drug, that is, its absorption, distribution, metabolism, excretion. And so what happens is if an individual is taking a particular drug, it will be exposed to that drug systemically through through a plasma exposure time curve as denoted on the slide, with x-axis being time and your y-axis being the drug plasma concentration. Now, if a patient takes another drug which prevents the metabolism of drug A, you will get a higher exposure of drug A in the presence of drug B. Now, this can be problematic because as you increase the exposure of the drug A, you may be getting to levels that may cause adverse drug events. Now, why do we look at P450s in particular? Now, Cytochrome P450s is a superfamily of membrane-bound heme thiolate proteins that metabolize a variety of substrates, both endogenous and exogenous. And of all the clinical drugs that are currently on the market, Cytochrome P450s are involved in the metabolism of 80% of those drugs. Now, more specifically, the isoform cytochrome P450 3A4, or CYP3A4, is the major player metabolizing at least 30% of those clinical drugs. And so it's important for us in the drug discovery all the way to development to understand, as we make new chemical entities, what potential risk we may have in inhibiting or altering the metabolism of already existing drugs. We don't want to make a drug that may potentially interact with another drug that our patient may be taking. And so what we need to do is understand and predict that risk as early as possible in order to select molecules that can make the best medicines. And so we do this through the process of in vitro to in vivo predictions. So in early discovery, we'll run a battery of different assays. The more complicated or kinetic ones are giving us an idea of potency as well as rates. And so we'll take those in vitro values and we'll incorporate them into either mechanistic static or dynamic physiologically based pharmacokinetic models, or PBPK models. Now, these models serve the purpose of understanding risk, and there are pros and cons to each. For the mechanistic static, we incorporate the aspects of the victim drug, that's FG, or the fraction that escapes gut metabolism, and FM, the fraction that's metabolized by that particular enzyme pathway. And we try to understand, in a worst case static scenario, what the potential interaction may be. While this is simpler to do, it is a worst case scenario, and it doesn't take into account the potential changes of your substrate as well as the inhibitor over time. And that's where dynamic PBPK modeling is an advantage, because it allows us to understand the temporal changes that exist with our substrate and inhibitor, as well as the physiological processes related to the enzyme. However, they require more sophisticated modeling. In either case, we generate what's called an AUC ratio, or a fold change in the exposure of the drug in the presence over the absence of a particular inhibitor. And so from that AUC fold change, we can then understand the potential risk and the severity of that risk, starting from weak to moderate to strong types of inhibition that we may potentially see with our new drug against the victim drug. Now, traditionally, we have not done a very good job at predicting drug-drug interactions for time-dependent inhibitors. We actually tend to over-predict the interaction, or in some cases, we have false positives, which means that in vitro, we find that we have a time-dependent inhibition signal, but when we go to the clinic, we don't see that time-dependent inhibition pan out. And so from the graph on the slide by Sue and colleagues in 2009, we can see that on the x-axis, we have our predicted drug-drug interactions using the human liver microsome fraction, and then the observed DDIs on the y-axis. And most of the points are shifted to the right, meaning that we're over-predicting a higher interaction compared to what's actually seen in the clinic. Now, over the years, people have postulated plenty of different ways of, of solving that problem in the disconnect. More recently, Phil Pula proposed that we could correct the fraction of the cytosolic concentration of drug in our calculations when scaling for the drug-drug interaction. When work is conducted in vitro in human liver fractions, we're just entering a concentration of the inhibitor. But in reality, with a cell, our inhibitor gets into the cell and then may be sequestered in lysosomes or other organelles, and the cytosolic concentration may be slightly lower. And so through in vitro work, you can actually determine that free cytosolic fraction and then correct the fraction of the drug that is actually exposed to the enzyme of interest. In the discovery space, Eng et al. Actually proposed establishing a boundary for an inactivation rate. Now, the inactivation rate is the rate of loss of the enzyme over time in the presence of the inhibitor. And what they proposed was that below a certain K observed, you would not likely be a drug-drug interaction in the clinic. Now, while this serves great for discovery, what you'll notice is that in many of the examples highlighted in the red boxes, the observed loss in vitro was much larger, but never panned out to a drug-drug interaction in the clinic, meaning that we may have potentially not moved that drug forward for risk of having a drug-drug interaction that didn't pan out. Now, the majority of the solutions all assume that the in vitro values are correct and that our ability to scale is what's incorrect. We need to find a different concentration or a different way about getting the inhibitor scaled for the interaction. But what if the in vitro values were the problem? So at a conference, we ran into a poster that was talking about substrate cooperativity. Now, so substrate cooperativity is not a new concept. It's existed for decades for P450s. But what this work demonstrated by Denisov and his group was that you have a different interaction with different substrates. Now, what they did is using lipid nanoparticle discs embedded with cytochrome P450 3A4, they looked at the metabolism of two substrates individually, progesterone and carbamazepine, and they also investigated the consumption of NADPH, which is the cofactor that P450s require to catalyze their reaction. And when they integrated both together in the same reaction, measuring the turnover of each of the substrates as well as the consumption, they found something quite interesting. For carbamazepine, what happened is that the production increased. So you were able to turn over the carbamazepine substrate faster. However, for progesterone, you actually had a decrease in that progesterone turnover. And interestingly, the NADPH consumption that would be predicted for both of those was substantially lower. And what they said was that the cytochrome P450 actually had a greater coupling efficiency, meaning that it could produce more by consuming less. And this is what's called heterotropic cooperativity, where one molecule alters the binding or kinetics of a chemically different molecule. Now, there is also what's called homotropic cooperativity, where the same molecule will alter its own binding and kinetics inside an enzyme. And this can both be positive or negative, where you can have increases or decreases in the rates. And this can occur at both the active site, so within the active catalytic site of an enzyme, as well as an allosteric site, which we'll get to in a few slides. Now, for cytochrome P450 3A4, there is the prototypical example of homotropic cooperativity, which is midazolam. Now, midazolam is metabolized by CYP3A4 to two metabolites, the 1-hydroxymidazolam and the 4-hydroxymidazolam. Now, at low concentrations of midazolam, we have a despairingly different production of the metabolites. We actually produce the 1-hydroxy in a much greater abundance compared to the 4-hydroxy. Now, as you increase the concentration of midazolam in your reaction, what happens is that you start to become more likely to form the 1 and 4-hydroxy. This is because the midazolam stacks inside the active site so that one midazolam is actually orienting the active site in a way that allows the midazolam that's coming in to either be at the 1 or 4 carbon equally. And so this is the homotropic cooperativity. But what does that look like in human liver microsomes in an enzymatic experimental setup? And so what we do is we incubate human liver microsomes with different concentrations of midazolam, and we look at the formation of the metabolites. So on the graphs on the slide, on the x-axis is your concentration of your substrate midazolam, and on the y-axis is the formation rate of the metabolite, on the left being the 1-hydroxy and on the right being the 4-hydroxy. Now, for the 1-hydroxy formation, we see something that's interesting. After a certain concentration of midazolam, you actually see a decrease in that formation rate of the metabolite. This is deemed substrate inhibition, whereby the substrate itself is inhibiting the formation of its own metabolism. Conversely, if we look at the 4-hydroxymidazolam, we see more of a Michaelis-Menten relationship, whereby at a certain point of concentration of midazolam, we reach a maximum rate of reaction, which is your Vmax. Now, the concentration at which half of Vmax occurs of the substrate is what's called your Km, or your Michaelis-Menten constant. And this is denoting the affinity of your substrate, of your enzyme, towards your substrate. But if we look at the raw formation of the 1,4-hydroxy, what we see is that at low concentrations of midazolam, we have a greater ratio of 1,4-hydroxy, meaning we're still making that 1-hydroxy more than we are the 4-hydroxy. But as the concentration of midazolam increases and that orientation occurs differently within the active site, we get more of a equal 1-to-1 formation of the 1 and 4-hydroxy. But what happens if you have something that is competing for the active site of or for CYP3A4? And so when we have reversible inhibition, we can perform this kinetic assay with the presence of different concentrations of the inhibitor. In this case, we're looking at ketoconazole, which is a reversible inhibitor of P450 Now, each line on the graph denotes the assay done in a different concentration of ketoconazole. And if we model then the enzyme kinetics, what we see is that our maximum velocity is unchanged, which for a competitive inhibitor, it's not changing the amount of enzyme that's there. And at high enough concentrations, we eventually reach and overcome the competition with the ketoconazole. But what we do see is that our Km, or the affinity for the substrate, appears to be shifted upwards, meaning that we need higher concentrations of midazolam to really get into that active site and overcome the ketoconazole that is present. Now, if we look at the 1,4-hydroxy in this scenario, what we see is that the low concentrations of midazolam, we've got a very much shift higher up in the ratio 1-to-4-hydroxy, meaning that what little midazolam is finding the enzyme is going to be predominantly conformed or converted to the 1-hydroxy as opposed to the 4-hydroxy. However, as you increase that midazolam concentration and you get high enough where the midazolam is starting to stack inside the active site, you recapitulate that homotropic cooperativity within CYP3A4, and you form the 1 and 4-hydroxy in an equal ratio. But this is interacting within the active site. And that's not ideal, because we want to understand what happens if we have allosteric modulation or changes with the enzyme where we're not interacting within the active site. And what we saw from Denisov's work is that when they modeled the results of the enzymatic work, the only way to explain the changes in both the production of the carbamazepine, the reduction of progesterone, and then the ideal coupling is that carbamazepine was predominantly in the active site, while progesterone would be positioned alternate to the active site at a different site. And if we go into the literature, we see that there's evidence of an allosteric alternate site than the catalytic site on CYP3A4. In 2004, Williams published an X-ray crystallography structure of CYP3A4 demonstrating the active site. And what they did is they also incubated the enzyme with progesterone. And what they found is that progesterone bound to a phenylalanine cluster outside of the active site. And in fact, that phenylalanine cluster is plays a role in both the initial substrate binding and overall conformation of CYP3A4. And so even though they incubated with different concentrations of progesterone, they were not able to capture progesterone inside that active site. It preferred the allosteric site. And there's been biochemical work to demonstrate either mutations in the phenylalanine will affect that CYP3A4 activity, but if you covalently bind progesterone to that phenylalanine cluster, you actually recover the activity and improve the CYP3A4 activity for the enzyme. Now, this work was done in lipid nanoparticles or recombinant enzymes, and not the typical subcellular fraction of human liver microsomes that we use when we're doing our translational work. And so the question was, what happens if we do the midazolam kinetics in the presence of the allosteric modulator progesterone? And so much like we did with ketoconazole, we repeat that experiment with different concentrations of progesterone, now instead of ketoconazole. And what we saw was something interesting. For 1-hydroxy, we do see that shift in affinity. Now our Michaelis-Menten constant for 1-hydroxy is a little bit higher than it was before, but we're not changing the Vmax, which may suggest that there's that competitive inhibition or competitive interaction within the active site. However, if you look at the other metabolite, 4-hydroxymidazolam, there is no change in affinity and no change in Vmax. If we were interacting within the active site, we would see an interaction in both the 1-hydroxy and 4-hydroxymidazolam formation. And so this gives us confidence that progesterone is binding to an allosteric site and altering the way in which the enzyme conformation is so that you're forming 1-hydroxy differently than you are in the absence of progesterone, but 4-hydroxy formation is unaltered. Now, what happens if you measure and look at the 1 to 4-hydroxy ratio? Well, in this case, we actually see that at low concentrations of midazolam, in the presence of progesterone, we recapitulate the homotropic cooperativity. So that change in the enzyme conformation, where now we're equally likely to generate the 1 or 4-hydroxymidazolam, but now at a much lower concentration of progesterone. Now, this was done in human liver microsomes, but we were glad to see that Denisov also performed a similar experiment later with a lipid nanoparticle disc, which is only with Cytochrome P450 3A4, and saw that with a low concentration of progesterone, you do recapitulate with the enzyme that 1 to 4-hydroxy ratio. And so now that we've shown that allosteric modulation can alter the kinetics, we ask the question, well, what happens if you apply this then to time-dependent inhibition? Can heterotropic cooperativity influence time-dependent inhibition in human liver microsomes? Where we're not interacting inside the active site, but actually altering the catalytic function of CYP3A4 by binding to that allosteric site. And we want to do this with human liver microsomes with mechanism-based inhibitors that we know are mechanism-based inhibitors. And there are several different mechanisms of irreversible time-dependent inhibition. Starting on the left, we have metabolic intermediate complex, and this is where a compound, once metabolized, the reactive species will then form a quasi-irreversible bond with the iron in the heme of the P450, essentially rendering it unable to catalyze other reactions. Two examples of this are clarithromycin and trolliendomycin. Another mechanism is the modification of the heme or destruction of the heme. Bifidil, once metabolized, will actually destroy the iron and remove it, so you no longer have the traditional P450 spectra, and essentially removing the heme doesn't allow for the catalytic reactions to occur, and the enzyme is inactivated. And finally, the covalent protein adduct. An example of this is raloxifene. Upon metabolism, that reactive species covalently binds to the enzyme. And when it does that, it actually blocks the active site so that no longer can a substrate go in. And so the enzyme is now inactive. Now, before we go to the results, I'd like to orient people to how we do these assays and the results and the data that we generate. So using human liver microsomes at a high concentration, we include an inhibitor as well as an NADPH cofactor to allow for the reaction and the conversion to those reactive species within the active site. Now, at different times, we'll take an aliquot of that reaction and then measure the activity of the enzyme through a substrate, in this case, midazolam. And what we see is that over time, we can then measure the activity that remains when the enzymes have been in the presence of an inhibitor that can inactivate it. We can then plot this over time, with the x-axis in this graph being time, and the y-axis is the percent of remaining activity of the enzyme compared to its starting point prior to the presence of the inhibitor. Now, each line represents the reaction in the presence of a different concentration of inhibitor. And you can see that as you increase the concentration of inhibitor, the slope of the line increases. So we have a time and concentration dependent effect by the enzyme. Now, the slope of that line is the observed loss in the presence of that particular inhibitor concentration. And you can replot this on a graph where the x-axis is your inhibitor and the y-axis is your observed loss of the slope of the line, and you see a relationship much like a Michaelis-Menten Michaelis-Menten graph, where now the maximum rate of activity is actually your K inact, or a first order inactivation rate constant, which tells you the maximum rate of which the inhibitor will inactivate your enzyme. And then your Ki is your irreversible inhibition constant, which is a measure of the affinity of your inhibitor towards your substrate. And so we can conduct this experiment in the presence of an allosteric modulator and see how this impacts the observed rate of change in the assay and eventually the kinetic parameters of Kinact and Ki, which are important when we then go to scale and predict our drug-drug interactions. So starting with raloxifene and mibifridil. When we incubate raloxifene or mibifridil with a low concentration of progesterone, so 10 micromolar, or a high concentration of progesterone, 100 micromolar, we saw some interesting results. Starting on the left with raloxifene, as we increase the concentration of progesterone, we see that there's really no change in the affinity of raloxifene towards the CYP3A4 enzyme. However, at the high concentration of progesterone, we actually start to see an effect on the inactivation rate, which is decreased in the presence of progesterone. Now, unlike competitive inhibition, where you would see no change in your Kinact, but you would see changes in your Ki, what we are seeing is akin to a non-competitive pattern that doesn't interact with the active site. In the case of mibifridil, we actually see that there's a small change at the low concentration of progesterone, but as we shift to the higher concentration, we're seeing both a change in Ki and Kinact, saying that the affinity of our inhibitor is decreased, our Ki is getting bigger, so the potency is getting lower, and the inactivation rate is decreasing. And so the efficiency of the inhibitor towards that enzyme is decreased. But the shift in these kinetic parameters, again, is not like competitive inhibition within the active site. It's actually akin to uncompetitive pattern of interaction outside of the active site. So again, bolstering our confidence that we're not competing with the time-dependent inhibitors for the active site and their ability in order to metabolize into the reactive species that causes their mechanism-based inhibition. But what about the metabolic intermediate complex compounds? So for clarithromycin and tropondiamycin, what we saw is they were a lot more sensitive to progesterone, where we could see some significant shifts at the low concentration of progesterone, 10 micromolar, and more significant shifts at the high concentration of 100 micromolar. Now, again, we see both shifts in potency as well as the inactivation, but the extent of which that happens is different depending on the inhibitor. Now, for MIC formers or metabolic intermediate complex formers, you can measure the formation of that MIC complex spectrophotometrically. So as the reactive species binds and creates that quasi-irreversible bond with the heme, you can measure a peak at 450 nanometers. And that's what's shown on the right. And so in different concentrations of progesterone, in the presence of an excess concentration of the inhibitor, you can see a formation of that MIC, but in the presence of progesterone, that peak decreases. Now, the argument can be made that if you add an excess of inhibitor and NADPH, that over time you'll eventually get back to that control amount of MIC formed, since there's still enzyme that could be that is potentially available to create that MIC. However, we did that experiment and we let it run for longer, giving excess NADPH as well as inhibitor. And what we found is that in the presence of progesterone, we form a smaller amount of MIC, but it's a new maximum amount of that MIC, meaning that the presence of progesterone is actually protecting the enzymes or having them in a formation that doesn't allow for the MIC to be formed. And so if we take then the kinetic rates for these and look at the efficiency, so how well the inactivation can occur by your inhibitor, you can calculate that inactivation efficiency much like you would your intrinsic clearance, which Vmax over Km. In this case, you're taking your maximum rate of inactivation, Kinact, over your Ki. And compared to the control, what we see is for all of our time-dependent irreversible mechanism-based inhibitors, there is a reduction in the inactivation efficiency in a concentration-dependent manner with progesterone, meaning that the inactivation decreases as you have progesterone on board. And that extent of reduction is different depending on the inhibitor that you have. However, the general sense is that the progesterone changes are driven by small decreases in your potency and much larger decreases in the inactivation rate. Now, in the publication that we have this work, and we also looked at the heterotropic cooperative molecule carbamazepine, which interacts with CYP3A4 as well, but it does so within the active site. And those changes are much larger changes in potency and less changes in the inactivation rate because it's competing within the active site. So not giving you a true change in the enzyme as the compound carbamazepine is actually interacting and competing with the ability for the inhibitors to enact their inactivation. Whereas progesterone is actually binding to the allosteric site and causing a different conformational structure of the CYP3A4, which is changing the efficiency of the inhibitor within the system. So at this juncture, we had found that progesterone is an allosteric heterotropic cooperative modulator of CYP3A4 in human liver microsomes, the subcellular fraction that we use for the work and discovery. And that heterotropic cooperativity actually can impact known mechanism-based inhibitors, both in their potency and in inactivation, but on a case-by-case basis. It's not a blanket way of how they interact. But most importantly, we still retained a positive signal. Even at high concentration of progesterone, we still see the time-dependent mechanism-based inactivation, which is a good thing because we don't want to alter conditions where we now have a false positive a true positive signal that is eliminated that we would then move forward and may potentially have risk later on. And so we maintain those true signals, and we're trying to understand the rates of inactivation better. Now, what we saw is that the mechanism of metabolic intermediate complex was more sensitive to progesterone. And so there is a class of compounds, the macrolides, which are notorious for forming MICs. And so we asked, well, do we see this same type of interaction in that class of mechanism across different molecules? And so we got a set of macrolides and we ran the conditions of no progesterone and 100 micromolar progesterone as a screen. And what we saw was we had varied response. In some cases, we saw reduction in our inactivation efficiency, both from Kinact and their Ki perspective. Now, we repeated clarithromycin and troleonomycin, as you saw earlier, and we also had erythromycin. In other cases, such as azithromycin, spiramycin, and roxithromycin, we actually eliminated that signal. You can see these are fairly weak K-observed rates. And so much like what Eng had proposed is that low rates of the observed loss of the enzyme are actually less likely to actually have the TDI. And in the presence of progesterone, we eliminate that signal. However, in the case of fidaxomicin, even at 100 micromolars of progesterone, we saw no effect. There was no change in the time-dependent inhibition caused by fidaxomicin, even in the presence of progesterone. Now, when we look at the formation of the metabolic intermediate complex, we see that we reduce the complex for the ones that we still see the TDI signal, whereas for those that are eliminated, we're no longer forming that MIC. Now, for fidaxomicin, we weren't able to observe an MIC formation. So it's likely that fidaxomicin's time-dependent inhibition is through a different mechanism than MIC formation. However, we knew that there was a clinical study for fidaxomicin that showed that there was no interaction. But when you dig into it, you see that the exposures for fidaxomicin are actually in the nanomolar range, so fairly low, where you can see from the graph on the slide, the fidaxomicin concentrations for the Ki, it's in the high micromolar range. So it's likely the TDI is possible, but you need to expose an individual to much higher concentrations of the drug in order to actually see it. And since the effective concentration or doses given for fidaxomicin are fairly low, no interaction is observed. And this highlights that TDI data or data in vitro is only part of that puzzle. You have to put it in context to how much you're going to give a drug and how much exposure a person will have for that particular drug. But 100 micromolars is a fairly high concentration, and it was more as a screen rather than a translation. While this is getting us lower inactivation efficiency, which helps in that overprediction, we wanted to really understand under what conditions can we truly replicate the in vivo interaction that we're seeing for time-dependent inhibitors. And so we set out to dial in that progesterone concentrations in vitro. And so we used clarithromycin and erythromycin and conducted that time-dependent inhibition kinetic assay in the presence of varying concentrations of progesterone. Now, we generated the Ki and Kinact, and there are physiologically based pharmacokinetic models for clarithromycin and erythromycin. Now, adjusting the model so that the exposure of the drug did not change regardless of the TDI kinetics, we ran simulations for clinical drug-drug interaction designs across a variety of different designs and for both the erythromycin and clarithromycin. And then we predicted, under the conditions of varying concentration of progesterone, TDI kinetics, what the AUC ratio would be, and then determined that there was a linear relationship, that as you increase the concentration of progesterone, you have a linear relationship in the loss of the AUC ratio. And across a variety of studies in 2 different inhibitors, we found that the presence of 45 micromolar progesterone was sufficient to replicate in vitro TDI kinetics that recapitulated across the board the in vivo inhibition. And so now with this modified optimized condition, the question was, does that work outside of just the clarithromycin or erythromycin across different mechanisms and across a variety of chemistries that exist? And so we scour the literature to find as many examples of time-dependent inhibitors in vitro that also had clinical drug-drug interaction studies. I found a total of about 46. And so we conducted the TDI kinetic assays either in the control conditions or in the presence of the optimized 45 micromolar progesterone conditions. And much like we've seen before, it's a mixed bag. In some cases, we could see that there was changes, either an increase in the Ki, which means that we are decreasing the potency, or an increase in the Kinact, means that we're making the inactivation rate higher. While in other cases, we would decrease the Ki, making it more potent in the presence of progesterone, or decreasing the activation rate, meaning that it's less inactive in its inactivation. But most interestingly, for a set of molecules denoted with the red X's, we eliminated that signal. And these were compounds that all did not have a drug-drug interaction in the clinic. So under these conditions, we're able to identify false positives through the in vitro human liver TDI assay that would have been positive signals, as you can see, fairly large inactivations for both of those molecules, or for those sets of molecules. And so we're able to do that, and we're altering the TDI kinetics. And so when we take those 2 pieces together, we see, again, a variety of response. In some cases, we actually increase that inactivation efficiency. In the presence of progesterone, the inactivation seems to be more efficient. Whereas in other cases, we actually decrease that inactivation efficiency for a much larger set of the molecules, meaning that we're less likely to inactivate that enzyme over time. And in some cases, we saw no changes in the inactivation efficiency, likely due to counterintuitive changes of Ki and Kinact, meaning if you have an increase in your Ki but a decrease in your Kinact, they sort of offset each other. And again, we have those molecules that were false positives are now eliminated in the presence of progesterone. Now, we've seen to have altered the inactivation of different enzymes, both in an increase or decrease or no change at all. And the question is, what happens if we then use these new values and try to predict the drug-drug interaction risk? And so we did this both for mechanistic static and dynamic modeling. And starting with the mechanistic static model, what we did is for the clinical victim substrates, so whatever the substrate was done in the clinic, we used its FG and FM. And using the model that was proposed by Singh et al., where we're using a concentration for the gut and the liver that have been optimized to give us better drug-drug interaction predictions. Now, this is using the average unbound plasma portal inlet concentration for the gut interaction and using the average unbound systemic concentration for the liver. So already a model that has been adjusted in order to try to get the best predictions possible using our in vitro human liver values. Now, on the graphs, you have on the x-axis the observed AUC ratio, and on the y is the predicted over observed AUC ratio. That solid black line is the unity, meaning we want to be as close to that as possible, meaning that we're hitting the mark. And then the dashed line is the twofold error. And if we look at the bias or the accuracy of our predictions through the average fold error and the precision of our predictions with the geometric mean fold error, we see that using data generated in vitro in the presence of progesterone, even though this is an already optimized model for the interaction predictions, we actually improve our accuracy and precision. So we're getting even better results. Now, in the case of PBPK modeling, there was a smaller subset of molecules that did have PBPK models. And the same story occurs where now when you integrate data generated in vitro in the presence of progesterone, you're getting better accuracy and precision of your predictions of drug-drug interaction. Now, these were already models that were fairly good. It seems like a small amount of improvement, but we're getting closer to the answer. And if you look at it from a perspective of if you didn't have any information, you would be selecting the molecules and having a better understanding of that risk. And what's important is how we categorize that risk. And so if we look at how the model predicts the types of categories that we would have, we actually have an additional improvement when we're using data generated in vitro with human liver microsomes in the presence of progesterone. Now, if we look at the true positives, which are defined by, we would have predicted an AUC ratio greater than the cutoff of 1.25, which denotes an interaction. And we also see that observed AUC ratio greater than 1.25. We pretty much are just spot on as the original model. Little improvement in the mechanistic static, but we're still capturing that true positive. Now, in the cases of true negatives, where we would predict an AUC ratio lower than 1.25 and the observed interaction is lower than 1.25 as well, we actually are capturing more of those cases with the data generated in the presence of progesterone, meaning that we're categorizing them as not a risk, whereas in the original models, they would have been categorized as a risk. And then finally, we have the false positives where we're predicting that there is going to be an interaction, and then in reality, there is no interaction when you get into the clinic. And there, where we've reduced those, we've moved them over to the true negatives in most of those cases. Most importantly is we have no false negatives where we would have predicted no interaction, and in reality, there was a reaction or an interaction in the clinic, which again is important. We don't want to eliminate the risk because for patient safety, we want to ensure that there is no interaction as we move that drug forward. And so the DDI risk predictions are improved in the presence of progesterone as compared to the traditional control way of which we do that. So to summarize, the addition of the optimal modified concentration of progesterone of 45 micromolars results in varied inhibitor-specific changes in the in vitro TDI kinetics across a variety of different chemistries. However, we know that we can improve the drug-drug interaction condition or the drug-drug interaction predictions under the conditions of having progesterone. And we improve the discrimination between true negatives and false positives, so categorizing that risk more appropriately when we use data generated with progesterone. But a lot of times we get the statement 45 micromolar progesterone seems like a high concentration. We're not circulating 45 micromolar progesterone in our bodies. And you're right, circulating concentrations of progesterone are not in that range. However, you have to put the presence of progesterone into context. Progesterone in the in vitro system, which is an artificial system, it's not in vivo, is meant to mimic holistic interactions of anything that may be interacting with that allosteric site in vivo. In vivo, we have protein-protein interactions that can bind to that allosteric site, as well as other endogenous substrates. In the figure on the right from Nakamura, we see that a variety of steroids that can also interact with CYP3A4, altering the turnover of different substrates. And so it's likely that there are many other things interacting with that allosteric site for CYP3A4 in vivo. And what we're trying to do is, in an artificial in vitro system, mimic that by having progesterone, which we know binds to that allosteric site, in the assay. The other thing to keep in mind is that circulating concentration is much different than tissue concentrations. The liver is a site of steroidogenesis and metabolisms, and so there's likely a much higher accumulation of steroids than what we see in the systemic circulation. So levels could be quite a bit higher than what we typically associate with the systemic circulation. So in conclusion, assay conditions matter. it's important to know what we're using when we start, what are our starting points when we do our modeling or our predictions. What we put in is very important, and what assumptions we make when we are generating this data is important. What I've hopefully demonstrated is that adding progesterone to human liver microsome time-dependent inhibition assays provides that mechanistic explanation to why we see that TDI disconnect, that the enzyme is in an artificial conformation that is lacking the interaction with its allosteric site, and that progesterone now provides. And importantly, we're able to eliminate false positive while maintaining true time-dependent inhibitions, meaning that we can move compounds forward if we see the loss of that TDI in the presence of progesterone that we may not have originally moved forward because of the TDI risk with that enzyme. Now, in the case of fidaxomicin, we've demonstrated that TDI is only part of the puzzles. There are other mechanisms involved, such as reversible inhibition, maybe induction. There's the change in the enzyme synthesis, as well as the changes in the substrate exposure and the inhibitor exposure with time. And that's the advantage of the PBPK modeling, which allows you to capture all those dynamic temporal changes in order to really understand and put into context what that TDI data from in vitro is telling you. And finally, while this condition actually is helping us, I think there's additional work that's needed to really understand some of those disconnects where we have the inability to use the human liver microsomes to really translate either metabolism or time-dependent inhibition due to the in vitro artifacts. And so there are many things that can alter an enzyme's function, including the pH, as well as the buffer constituents and what you have in your buffer. And so it's really important to try to find an in vitro condition that mimics the in vivo environment as much as possible so that we're starting with a really reflective in vitro value. And then we can then start to get at those other solutions of when we're missing the mark, what are we truly missing? Is it a different concentration of inhibitor that's being seen at the site of action or something else? With that, I'd like to acknowledge my collaborators, of which none of this would have been possible. And if there are any questions, I'm happy to take them.
Thanks, Luke. That was a really great presentation. Yeah, the session's open for any questions if anyone has any. Yeah, so there's a couple of questions perhaps coming through. Armin, do you mind unmuting and then asking a question?
Hello, yeah, my name is Armin. Thank you for this insightful talk. I really wanted to ask about the specificity of such interactions. We're thinking here about binding to the active site, to the allosteric site, and so on and so forth. But the affinities are fairly low. We're talking micromolars, tens of micromolars, if even more. How confident we are that the interactions at such fairly low affinities actually all happen at the same place in the same active site, that they are not just arbitrary patches on the protein surface that just happen to be perhaps individual for each drug, and they all have different influences, effects as a result.
So if I understand your question, are you referring to the inhibitors themselves or to the allosteric modulator with their
Let's say allosteric modulators.
Gotcha. Yeah, that's a good question. So from the work from Williams, despite their best efforts, they weren't able to capture progesterone within the active site. And also, if you look at the enzyme kinetics for progesterone, its Km is actually fairly high. It's close to about 100 micromolar. So at 100 micromolar, you're reaching half of the maximum activity of the enzyme CYP3A4 towards progesterone. So it prefers to bind to the allosteric site over the active site. Now, it doesn't mean that there isn't potentially interactions within the active site, but given that its affinity is fairly low, it's most likely less of an interaction than what would be for something that would have a higher affinity for the active site of CYP3A4.
Okay, thank you.
Iman, did you want to raise your point?
Yes, please. Thank you. Thank you for the presentation, Luke. It's very nice. I just have a question regarding the progesterone specifically, because it's not only an allosteric modulator, but it can also an inducer for the CYP3A4 expression. So I think this might result in the mismatch between the in vivo and the in vitro results, because in the in vitro, especially with human liver microsomes, you will not see such effect of the induction, which in vivo might lead to reduction in the ratio of the AUC.
Yeah, so you're referring to if induction is also a mechanism of interaction for an inhibitor or an inducer or perpetrator?
For the CYP3A4 itself as an enzyme. So it will increase the enzyme, so the effect of the inhibitor will be lower than expected. I mean, the AUC ratio will reduce due to the induction, not due to the allosteric inhibition only.
Yeah, so all this work focuses only on time-dependent inhibition, and you're absolutely right that there are multiple mechanisms, which is part of the point on the last slide, that it's only part of that puzzle, right? So if you have time-dependent inhibition, but you also have an inhibitor that also causes induction, you're correct that you may not see that signal because it washes out due to that. And so in there, it's important to use a dynamic simulation where you can incorporate both the induction as well as the time-dependent inhibition to see whether or not they offset each other. Whereas in the mechanistic static, you can, but the FDA does not recommend that you make those drug-drug interaction predictions using inhibition and induction in the same calculation. But for the human liver microsomes, you won't see that induction because it's the enzyme. It's not in a cellular system where you could induce that. So it's really specific to the time-dependent inhibition, but you're absolutely right. It's important to see all the different interaction potentials and put them together to really understand if you see that.
Yeah, thank you.
Are there any other questions? So, Luke, is this something now that you would routinely do assays with, or do you still have to do things back to back with and without this progesterone?
Yeah, that's a great question, Oliver. So we would do it with both, right? We would do it traditionally as the control, and then we would also do it in the presence of progesterone, and that would help you at least early on to determine, if that signal goes away, then, it's a false positive, and so you're likely to move that molecule forward. But if it continues, then you can start to understand, well, is that a molecule that I want to take forward? Is that a risk I'm willing to take? Or is there another molecule that doesn't have that risk, or that risk is eliminated? So it's helping in the selection of the molecules, but until there's greater confidence across a much, much larger N of chemistries and molecules, we're running both.
Yeah, I understand. That sounds reasonable. But yeah, I guess you're working towards that, building that confidence in it. I'll just take a question from Dan. Would you like to unmute?
Are we unmuted? Can you hear us right now? Hello?
Hello? You're quite quiet, but if you can speak into the mic, that would be great.
Hear us?
Yep.
Okay. I have a question. Progesterone binds at an allosteric site that has a bunch of phenylalanines. It occurs to me that there could be other drugs that mimic that effect. Are there known allosteric modulators of 3A4 that are drugs?
That's a great question. Well, so we do see heterotropic cooperativity, which is so allosteric modulation. It can be defined both as the active site and the allosteric site. So it's a site that's alternate to the catalytic site. So it can still be within the active pocket. And so carbamazepine is an example of that, where it will actually cause that cooperativity within the active site as opposed to binding to that allosteric site. regarding compounds that bind to the outside allosteric site, I don't know of any drugs. I know there is evidence with other substrates for CYP3A4, so testosterone has also been shown to bind to the allosteric site. So other steroids or endogenous molecules, but in terms of drugs that bind to the allosteric site, I'm not aware of any.
Okay. Yeah, because the other examples like midazolam and carbamazepine interact inside the active site. It occurs to me there's probably things that we don't know about that bind to the allosteric site and may have effects.
Yeah, absolutely. And we don't routinely screen or look at data in order to really differentiate where it's binding. It's more of a conversion.
Yeah. It's not something you'd look for, but it may present at some point, but just curious. Nice talk.
Thank you.
Yeah, there's one more question, and it's from Ankita. Did you want to ask a question?
Yeah. Can you hear me, Oliver?
Yep.
Yeah. So my question is, like, generally when we conduct in vitro experiments, we generally take healthy human tissue. But if in a disease state or due to an effect of a drug, if you have fluctuation of all this endogenous substance that you mentioned, like progesterone, estradiol, or testosterone, would you also expect a disconnect from in vitro to in vivo? And possibly it can be in both direction. You can also expect more interaction than observed in vitro, depending on how your progesterone, estradiol, testosterone levels are fluctuating in response to that disease or a drug. Have you seen any examples of this, or does that sound like something possible?
That is a fantastic question. And yes, it is possible that circulating concentrations may alter that. And there's been some work in the early 90s, Larry Winker and folks from Amgen had looked at the changes in postmenopausal and premenopausal women where steroid concentrations are different, and looked at the CYP3A4 activity. And you can see differences depending on the circulating concentrations. So in disease states where that may be altered, or in normal hormonal reproductive states, you may actually have a difference in how your enzymes are functioning. So it's definitely something to keep in mind, especially for disease states, but that is possible.
Thank you.
Okay. I don't think there are any more questions coming through. I'd just like to thank Luke again for your fantastic presentation and for agreeing to present to us today. There is a new webinar being put together, and this is on the PPBM template, which will be coming soon. So there'll be registration for that, and that will be in the next month or so. But otherwise, thanks again for joining us, and speak to you next time.
Thank you for the opportunity. Thank you, everyone.
Take care. Bye.
Bye.